So first of all, thank you for giving me the opportunity to talk about a method we have developed recently. It's about the detection and quantification of introgression using Bayesian inference based on conjugate priors. And this is joint work with Joel Capon. So what is introgression? It refers to the flow of genes between species and is considered to be a major force structuring the evolution of genomes, which potentially can provide raw material for adaptation. And the last years have seen quite a lot of methods in the field, which can be applied to whole genomes or on smaller genomic windows to quantify and to detect introgression. A general class of method utilize patterns of single nucleotide polymorphisms for taxon trees, so where you have three populations and one outgroup. And the most popular family of statistics are the ABA-BABA types of statistics, and we will concentrate on this type of statistic. So here we can see a species tree. And assuming we have an infinite sites model, then every mutation creates a single nucleotide polymorphism. In panel B is shown a typical example of introgression where population 3 and population 2 are sharing the mutant allele B, resulting in the overall pattern ABBA. But this pattern, in fact, can also be created by neutral signals like incomplete lineage sorting. Here in panel A, you see that ELS can produce basically the same pattern, ABBA, and also other patterns can easily be created by incomplete lineage sorting. To detect introgression, the Patterson's D statistic was proposed, which basically subtracts the counts of ABA patterns and the BABA patterns, which refer to introgression. And in the denominator, it summarizes the patterns. And it's really cool that this can be used as a statistic because the denominator captures also the neutral signal due to incomplete lineage sorting. So on a whole genome where a lot of such signals occur, this will automatically be in the denominator. And problems with Patterson's D are that it's not designed for smaller genomic windows. It's not directly proportional to the fraction of introgression. And there are a lot of false positives due to low genetic diversity, which suggests to also consider distances on a full taxon tree. This is basically what we did 2019 with the publication Estimates of Introgression as a Function of Pairwise Distances. And there we took a twofold approach where we reformulated the Patterson's D in terms of distances, and to this end resulting in this formulation where P xk is the mutant allele, in this case B, frequency in population X at variant site K, and the D are the distances between the populations. And from this formulation, we can easily go back to the pattern version, and it turns out that our statistic is quite similar to the Patterson's D, but this special term is added in the denominator 2 times BBAA. And interestingly, BBAA is the species tree pattern, which means we add kind of neutral signal into the denominator. Results show that this quite accurately quantifies the fraction of integration. So on the x-axis, we see the fraction of integration, the statistics values, and the red horizontal lines are the real fraction of integration. And you see that our method, df, distance fraction statistics, quite accurately quantifies integration, whereas Peterson's D is quite inflated. So in this case, this was gene flow from population 3 to population 2, the other way around, from population P2 to P3. It turns out that all methods, or the DF and the competing method FD, quite underestimates the fraction of integration. Again, Peterson's D is inflated. Yeah, and in this case, I think we applied 10 kb windows, so quite small genomic windows. So some pros about the DF statistics: it quantifies integration more accurate compared to the state of the art. It is robust to the time of gene flow, which we will see later again. And it provides a symmetric solution, so it can quantify integration in both directions, from P3 to P1, but also from P3 to P2. What is not yet perfect with the method is that it does not take into account the number of variant sites within a genomic region. And so we have developed a Bayesian framework, which quite naturally takes this into account. So we call it the DFBF approach. It's a Bayesian model selection approach for the detection and quantification of integration. It quantifies integration with the inferred inbuilt theta parameter, and at the same time enables weighting the strength of evidence for integration based on Bayes factors. So the main idea is that we define two models, M1 and M2, where M1 captures the scenario where taxon population 3 and 2 are sharing alleles, and M2 where population 3 and 1 are sharing alleles. So, and for the first try, we applied a simple binomial model with beta conjugate priors. So you see that the model includes two parameters, and the observations are reflected here. And that's basically the terms we saw in the already published DF statistics. So, for instance, P2 times D13 reflects the ABA plus BBAA pattern, and P1 times D23 captures the BABA plus BBAA pattern. So by applying this method, it just looks at each SNP and simply update the prior distribution, like you can see here. So the prior is initially set to the average population size of P1, P2, and P3, and then the parameters get updated accordingly with these distances on a full taxon tree. So this is a corresponding likelihood function with the gamma distributions, and you see that the framework includes this df theta parameter, which can be used to quantify integration. And the Bayes factors basically give us a hint how confident we are about this value here. So Bayesian factors are calculated as seen from this slide, and we interpret the values according to Jeffrey's table, which says a Bayesian factor of 1 means no evidence. Between 1 and 3, it means there's some signal. 3 to 10, there's moderate evidence, and then from 10 it starts to get strong. So simulation results indicate that the model parameter df theta quite accurately quantifies introgression, similar to the df statistics we have proposed. But we now, with this framework in hand, we also have these Bayesian factors for each fraction of integration, and it starts with a fraction of 0.9 to get a strong evidence. So the same simulation, but now varying the time of gene flow, which I said was really a good thing about the DF statistics. And you see this basically. We varied the time of gene flow on the x-axis and set the fraction of introgression to 0.5. You see the horizontal line here. As you can see, competing methods like the Patterson's D in blue or the FD statistics in green are quite affected by the time of gene flow, whereas df and the model parameter df theta are quite robust. And again, here on the right side, the corresponding Bayesian factors. So to summarize, we propose a new method DFBF, which quantifies introgression, is equally or more accurate than the DF statistics, and at the same time enables quantification of the strength of evidence for introgression based on Bayes factors. And we have implemented the methods within a R package pop genome, so researcher can apply these methods to whole genomes, sliding windows or feature-based windows like scanning genes or so on. Yeah, so thanks a lot.